This is podcast 20. So we had been talking about photosynthesis and the light-dependent reactions. Now we're going to talk about the non-light dependent reactions, sometimes called the dark reactions, formerly known as the Calvin cycle, not formerly, but formally, named after Melvin Calvin, who worked out this cycle. So the reason that I'm not crazy about the name the dark reactions is because it confuses people. These reactions don't happen in the dark. They depend on the products made from the light reactions. They depend on ATP. They depend on NADPH. But they don't happen in the dark because as soon as you stop making these things in the light reactions, then you also stop the quote-unquote dark reactions, the Calvin cycle. The Calvin cycle happens in the stroma, so inside the two outer membranes, outside the innermost thylakoid membrane. And the key to understanding the Calvin cycle is to follow the carbons, and it can be kind of confusing because you got to pay attention to how many molecules of each thing do we have. So 3 here, 6 here, 6 here, and so on. Versus and the other pathways we've been talking about just following one molecule throughout. So we're going to start with a 5-carbon molecule that is phosphorylated twice on its number 1 carbon and its number 5 carbon. So 2-phosphate is called bisphosphate, and this is called ribulose, 1,5-bisphosphate, and there's 3 of these molecules. We're going to add 3 CO2s. Where do they come from? They come from the atmosphere. They enter the plant through leaf openings called stomata. And the CO2 from the atmosphere is what gets built into the body of the plant. So these 3 5-carbon molecules plus 3 CO2s yield 6 3-carbon molecules. So 3 5-carbon molecules, that's 15 carbons, plus 3 more carbons in CO2, that's 18 carbons. 6 times 3 carbons is actually 18 carbons. So that's how you follow the carbons. In this first reaction, there's actually a 6-carbon molecule that is formed first, but it breaks down immediately, and they don't even list it on the side diagram, but you may see it on another Calvin cycle diagram that you get a 6-carbon molecule, and then you break down into this 3-carbon molecule. Then the terminal phosphate from ATP is added onto this 3-carbon molecule, and it becomes 1,3-bisphosphoglycerate. Then the energy from NADP is harvested. This energy was made because NADP is the terminal electron acceptor in the light reactions. So the NADPH is now oxidized to NADP, and that energy is used to convert 1,3-bisphosphoglycerate into glyceraldehyde 3-phosphate. So you've seen this before. It was called glyceraldehyde 3-phosphate in glycolysis right here. And so this is basically half a glucose molecule. And so if you go around this cycle twice, there's 6 of these that are made. Each time you peel 1 away. So you go around this twice, you peel 2 of these away, and you can make a hexose, a 6-carbon sugar, out of 2 3-carbon sugars. So if there's 6 of these 3-carbon molecules, and then you took 1 away, there's 5 left. Okay. The 5 left are 5 3-carbon molecules that gets converted into 3 5-carbon molecules. So 5 times 3 carbons is 15 carbons. 3 times 5 carbons is 15 carbons. Okay, ribulose 5-phosphate, and then you're back at the beginning, ribulose 1,5-bisphosphate. The enzymes involved in this reactions, and this is a pretty simplified version of the Calvin cycle. CC1, the first reaction, is ribulose bisphosphate carboxylase. We're going to skip the oxygenase for a second. The short name for this is Rubisco. It is a super abundant enzyme, and it's an interesting enzyme. We'll talk more about that. CC2 here, the second reaction, phosphoglycerol kinase, CC3, glyceraldehyde 3-phosphate dehydrogenase, and CC4, phosphoribullokinase, BRK. Rubisco is a large and very well studied protein because it's so important for life on the planet. There is more of this protein on Earth than any other single protein, that's an estimate, but 40 million tons estimated, or 15 pounds of this protein per person, because it's in all photosynthesizers. It's a very large complex protein, so large that you would need a specialized gel if you're running SDS page, for example, to even see it, because it's so big it would be hard to get it to enter the gel. Besides all these amazing things about Rubisco, it's not a very efficient enzyme in that its ability to fix CO2 is not that efficient. And there's a competing activity of this enzyme which makes it inefficient. So back here, we said that ribulose bisphosphate is both a carboxylase and an oxygenase, and this oxygenase activity is the activity that makes it inefficient. So in the presence of high concentrations of oxygen, it'll add an oxygen onto ribulose 1,5-bisphosphate and make a product that is a dead end for the Calvin cycle. And this really shuts down the efficiency of Rubisco. And there has been serious effort to try to genetically engineer Rubisco in order to make it more efficient and to eliminate this oxygenase activity, and that has not worked. There have evolved different systems that are more efficient, however. These are so-called C4 systems, because the first carbon intermediate that you can isolate from these systems is a 4-carbon compound, as opposed to Calvin cycle, where the first thing you can isolate is a 3-carbon compound. Okay, in C4 plants, what happens is essentially they concentrate the CO2 away from the atmospheric source, away from where there could be high concentrations of oxygen, and then release it in a place that is most favorable to the Calvin cycle. So CO2 is brought in through these pores and then converted into or fixed onto oxaloacetate by phosphoenolpyruvate carboxylase, PEP-C, and this has a high affinity for CO2, higher than Rubisco does, and is also not affected by high concentrations of oxygen. Then that oxaloacetate is converted into malate by NADPH-dependent malate dehydrogenase. Then that malate is physically moved to an adjacent cell, an adjacent cell, anatomically far away from where the CO2 comes in. And then the CO2 is removed from the malate, allowing CO2 to build up to high concentrations in a place that is favorable now for Rubisco to do its thing and fix carbon with more of a carboxylase activity than an oxygenase activity. So the strategy is to just build up high concentrations of CO2 so that the equilibrium is favored toward the carboxylase activity and away from the oxygenase activity. And in fact, these plants are more efficient than C3 plants. C4 plants are things that strive have real success in arid environments and really hot environments. Some C4 plants are ones that you know well, like corn, for example, and a lot of weeds are C4 plants. There are other pathways that also get around this oxygenase problem of rubisco. Okay, here's an anatomy picture to give you an idea of what is happening. So the CO2 is coming up in through these stomata. A single one is called a stoma. And then you have mesophyll cells with pyruvate, the PEP-C, phosphoenolpyruvate carboxylase activity in these cells. And then those cells are fixing the CO2 and then physically moving through malate those molecules into bundle sheath cells, which then releases the CO2. So you can build up a high concentration of CO2 in these cells and favor the equilibrium toward fixing carbon instead of the oxygenase activity. Okay, here's an H5P question for you.